Reactions of carbohydrates. Monosaccharides react with acid to form furfural, and these furfurals in turn condense with phenol to form violet ring. Now this is a basis of Morley's test, and this is mainly used for identification of carbohydrates and is a general test for carbohydrates used in lab. Next is alkali. When carbohydrates with free sugar group react with mild alkali, they form NADOls, and these NADOls are powerful reducing agent. They reduce cupric ions to cuprous ions, forming colored precipitate. Now this is the basis of Benedict's test. Now Benedict's test is also called as semi-quantitative test. This is because we can quantify the different colored precipitate, and this is mainly used in identification of reducing sugars in laboratory. So green precipitate, formation of green precipitate indicates 0.4% of reducing sugar in the patient sample. Yellow precipitate indicates 1%, orange precipitate indicates 1.5%, and red precipitate indicates 2% of reducing sugar excreted in the urine sample. Treatment reaction with the strong alkali results in formation of caramelization. Next is oxidation. Oxidation of first carbon atom of carbohydrate results in formation of aldonic acid. So glucose from gluconic acid, mannose forms mannonic acid, galactose from galactonic acid. Oxidation of last carbon atom of carbohydrates form uronic acid. For example, mannose forms mannuronic acid, galactose from galactouronic acid, and glucose from glucuronic acid. Formation of glucuronic acid is of great biological importance because glucuronic acid formation is used in phase 2 detoxification reaction, where the toxic metabolites or the harmful chemicals like drugs, antibiotics are detoxified in our body by conjugation with glucuronic acid, followed by excretion in urine by the renal system. Now glucuronic acid is also used in the synthesis of glycosaminoglycans like hyaluronic acid and chondroitin sulfate. Next reaction is reduction. Carbohydrates are reduced to alcohols. Example include fructose is reduced to mannitol, and this is used in the treatment of cerebral edema, whereby osmotic effect mannitol decreases the edema that is present in the cerebral cortex. Glucose is reduced to sorbitol, and this has been implicated in the development of diabetic cataract and diabetic neuropathy as a complication of long-term type 2 diabetes mellitus. Galactose is reduced to dulcitol, and this dulcitol has been implicated in the development of congenital cataract among children with the deficiency of galactose metabolism. Next one is formation of amino sugar. Now these amino sugars are formed by replacement of OH group at second carbon atom of glucose by amino group. So the example include glucose gets converted to glucosamine, galactose gets converted to galactosamine, mannose get converted to mannose-amine. Now these amino sugars are component of glycosaminoglycans, which are in turn they are involved in formation of extracellular matrix. Next is formation of deoxy sugar, where there occurs removal of hydrogen atom from second carbon atom of glucose. Now these deoxy sugars are used in formation of nucleic acid, especially DNA. Next is formation of glycosides, where the hemiacetyl group of glucose or any carbohydrate condense with phenol to form glycoside. So one good example is condensation of galactose with digitogenin to form digitonin, and this is used as cardiac stimulant, but with better availability of so many newer drugs now, so this digitonin is no longer used currently. Next is formation of esters, where the sugars combine with phosphate to form esters, and for example include the glucose 6-phosphate, fructose 6-phosphate, fructose 1,6-phosphate. All these are required for activation of sugars to enter different metabolic pathways. So the last reaction is formation of ozone, and this property is utilized for differentiating disaccharides in the laboratory, where lactose forms ball pattern shaped ozone crystals and maltose from sunflower shaped ozone crystals. So this was all about reaction of carbohydrate.